Listening Practice, Chapter 1, Introduction to Modern Biotechnology. Biotechnology has evolved from ancient practices like fermentation to a multidisciplinary powerhouse reshaping life in the 21st century. No longer confined to labs, it now intersects with artificial intelligence, climate science, and ethics, driving solutions for global challenges, from curing diseases to feeding a growing population sustainably. This chapter frames biotechnology's vast scope, its transformative sectors, and the collaborative, and sometimes contentious, roles of public and private institutions in advancing this field. 1.1 The scope of biotechnology in the 21st century. Modern biotechnology transcends traditional boundaries, merging biology with engineering, data science, and materials innovation. Its scope includes genetic engineering, CRISPR-Cas9, base editing, and synthetic biology enable precise manipulation of DNA across organisms. Cellular agriculture, lab-grown meat and dairy products, like those from *Upside Foods*, bypass traditional livestock farming. Environmental solutions, engineered microbes digest plastic waste, while algae-based systems capture carbon emissions. Human augmentation, brain-computer interfaces and gene therapies blur the line between treatment and enhancement. The field's interdisciplinary nature fuels breakthroughs like mRNA vaccines, COVID-19, and *bold threads synthetic spider silk, proving biology itself is becoming a programmable technology. 1.2 Key sectors, healthcare, agriculture, environment, and industry. Biotechnology's impact spans 4 critical domains. 1. Healthcare, personalized medicine. CAR T-cell therapies target individual cancer mutations. Diagnostics, AI-powered tools like *CRISPR-based Sherlock detect pathogens in minutes. 2. Agriculture, climate-resilient crops, drought-tolerant GMO maize and nitrogen-fixing microbes reduce farm inputs. Vertical farming, lab-engineered crops thrive in controlled environments, slashing water use. 3. Environment, bioremediation, bacteria like Idionella sacchensis break down PET plastics in landfills. Biofuels, algae-derived jet fuel cuts aviation emissions by 70%. 4. Industry, biofabrication, companies like Ecovative grow mycelium-based packaging as a styrofoam alternative. Enzymatic manufacturing, designer enzymes from novozymes replace toxic chemicals in textiles. 1.3 Public versus private sector contributions. The biotechnology revolution thrives on a symbiotic, and often competitive, relationship between public and private entities. Public sector funds foundational research, for example, NIH grants for CRISPR safety studies. Establishes regulatory frameworks, for example, USDA's 2023 approval of lab-grown chicken. Addresses global inequities, for example, WHO's mRNA vaccine hub in South Africa. Private sector, drives rapid commercialization, for example, Moderna's mRNA platform pivoting from cancer to COVID-19. Invests in high-risk ventures, for example, Memphis meets $186 million funding for cultured meat. Faces criticism for patent hoarding, for example, CRISPR intellectual property battles. Case study in collaboration, the BioMADE Institute, a U.S. government-funded consortium, partners with startups like Ginkgo Bioworks to scale bioindustrial innovations, blending public goals with private agility. Chapter 1 Summary. Modern biotechnology is a tapestry of science, ethics, and economics. From editing genes to reimagining food systems, its applications are as diverse as its stakeholders. Yet, its promise hinges on balancing profit-driven innovation with public good, a tension explored throughout this book. The following chapters delve into specific breakthroughs, such as lab-grown meat and synthetic silk, to reveal how biotechnology is rewriting the rules of life itself. Chapter 2: Historical Foundations of Biotechnology. Biotechnology's roots stretch back millennia, but its modern form emerged through groundbreaking scientific discoveries, ethical reckonings, and technological leaps. This chapter traces the field's evolution from ancient practices to today's precision tools, highlighting pivotal moments that transformed biology into an engineering discipline, and ignited debates that still shape its trajectory. 2.1 From fermentation to CRISPR, a timeline of breakthroughs. Biotechnology's history is a story of humanity harnessing biology for survival, innovation, and control. 10,000 BCE, early fermentation practices, bread, beer, cheese, mark the dawn of unconscious biotechnology. 1676, Antony van Leeuwenhoek observes microorganisms under a microscope, revealing the microbial world. 1928, Alexander Fleming discovers penicillin, launching the antibiotic era and industrial microbial cultivation. 1953, Watson, Crick, and Franklin unravel DNA's structure, unlocking the molecular basis of heredity. 1973, Herbert Boyer and Stanley Cohen pioneer recombinant DNA technology splicing genes across species. 1983, Kary Mullis invents PCR, polymerase chain reaction, revolutionizing DNA amplification and diagnostics. 2012, Jennifer Doudna and Emmanuel Charpentier develop CRISPR-Cas9, enabling precise, programmable gene editing. This timeline reflects a shift from accidental discovery to deliberate design, setting the stage for synthetic biology and beyond. 2.2 The role of genetic engineering, 1970s present. The 1970s marked biotechnology's transition from observation to intervention. Recombinant DNA breakthroughs, 1978, Genentech produces synthetic human insulin using engineered E. coli, replacing animal-derived insulin. 1982, the first GMO, genetically modified Pseudomonas bacteria, is approved to combat frost on crops. Agricultural Revolution, 1994, the Flav-Save tomato becomes the first commercially grown GMO food, engineered for delayed ripening. 2000s, BT cotton and golden rice, vitamin A enriched, highlight GMO's potential, and controversies. Human Genomics, 2003, the Human Genome Project completes its draft, accelerating gene therapy and personalized medicine. 2010s, CRISPR democratizes gene editing, enabling labs worldwide to modify organisms with unprecedented ease. Genetic engineering's legacy is dual-edged, it birthed life-saving therapies and fueled debates over playing God. 2.3 Ethical debates and the Asilomar Conference. Biotechnology's power to reshape life has always sparked ethical scrutiny. The Asilomar Conference, 1975, scientists self-imposed a moratorium on recombinant DNA research, fearing unintended consequences. For example, engineered pathogens resulted in containment guidelines, for example, biosafety levels, and federal regulations, setting a precedent for responsible innovation. Modern controversies, GMO backlash, public distrust of frankenfoods, led to strict labeling laws in the EU and activist movements like Greenpeace's anti-GMO campaigns. Human germline editing, the 2018 CRISPR-edited babies scandal, Hejian Kui, reignited debates over ethics, equity, and oversight. Synthetic biology. Critics warn of biohacking risks and ecological disruption from engineered organisms, for example, gene drives. Legacy of Asilomar. The conference established science's capacity for self-regulation but also revealed gaps in addressing societal fears. Today's challenges, like AI-driven biodesign and corporate control of genetic IP, demand new frameworks for global governance. Chapter 2 Summary. From fermenting beer to editing human embryos, biotechnology's history is a testament to human ingenuity and fallibility. The Asilomar principles of caution and transparency remain vital as CRISPR, synthetic biology, and AI push the field into uncharted territory. As later chapters explore lab-grown meat and microbial factories, these historical lessons underscore a recurring question: how do we balance innovation with responsibility in a world where biology itself is a tool? Chapter 3 Cellular Agriculture, Lab-Grown Meat 3.1 The science of cultivated meat. Lab-grown meat, or cultivated meat, is produced by cultivating animal cells in controlled environments, bypassing traditional livestock farming. 3.1.1 Cell sourcing and scaffolding techniques. Cell sourcing. Starter cells are typically obtained via biopsies from living animals, for example, muscle or stem cells. These cells are then multiplied in labs. Companies like Upside Foods use pluripotent stem cells for their versatility. Scaffolding. Scaffolds provide 3D structures for cells to grow into tissues. Materials include plant-based polymers, for example, cellulose, or biodegradable synthetics. Example: Eat Just, Good Meat, uses soy protein scaffolds to mimic chicken texture. 3.1.2 Nutrient media and bioreactor design. Nutrient media. Cells require a nutrient-rich broth, amino acids, sugars, growth factors. Fetal bovine serum, FBS, was traditionally used but is being phased out for plant-based alternatives. Cost hurdle. Media accounts for 50–80% of production expenses. Bioreactors, large-scale tanks, up to 10,000 liters, regulate temperature, oxygen, and pH. Design challenges. Ensuring uniform cell growth and preventing contamination. 3.2 Regulatory milestones. USDA approval, 2023. In June 2023, the USDA granted Good Meat and Upside Foods approval to sell cultivated chicken in the US, marking a watershed moment. 3.2.1 Case study. Good Meat and Upside Foods. Good Meat partnered with regulators to address safety, labeling, and facility inspections. First served in Singapore, 2020, before US approval. Upside Foods focused on scaling bioreactor tech and reducing media costs. Collaborated with Michelin star chefs to refine product appeal. 3.2.2 Global regulatory landscapes. EU, strict pre-market approvals under the Novel Food Regulation. Startups face lengthy reviews. Asia, Singapore leads, first to approve Good Meat in 2020, while China invests heavily in national cultivated meat initiatives. Middle East, Israel and UAE offer grants and tax breaks to attract startups. 3.3 Environmental and economic implications. 3.3.1 Reducing livestock emissions. Lab-grown meat could cut greenhouse gas emissions by up to 92% and land use by 90% compared to beef. Oxford study, 2021. Caveat: energy-intensive bioreactors may offset benefits if powered by non-renewables. 3.3.2 Cost challenges and scalability. Current costs, $10–20 per cultivated chicken breast, down from $330,000 in 2013. Scalability barriers, bioreactor capacity limitations. Dependency on affordable, FBS-free media. Projections, industry aims for price parity with conventional meat by 2030. Key takeaways, lab-grown meat combines biotechnology and food science to address sustainability and ethical concerns. Regulatory wins in 2023 signal growing acceptance, but global frameworks remain fragmented. Environmental benefits hinge on clean energy adoption and cost reductions. Next steps, partnerships, for example, Cargill investing in cultured fat startups, and tech innovations, AI-optimized bioreactors, will drive scalability. Public education is critical to overcoming the ick factor. Chapter 4, Synthetic Biology and Biofabrication 4.1 Engineering microbes for novel materials. Synthetic biology enables the design of microorganisms to produce materials with unprecedented precision, sustainability, and functionality. 4.1.1 Case Study, Bold Threads Microbial Spider Silk, 2023, Technology Overview. Bold Threads engineers yeast, Saccharomyces cerevisiae, with synthetic spider silk genes, derived from orb weaver spiders. Fermentation, yeast are grown in bioreactors, secreting silk proteins into nutrient broth. Purification and spinning, proteins are extracted, dissolved in solvent, and spun into fibers mimicking natural silk. 2023 milestone, Bold Threads scaled production for commercial partnerships, including a high-profile collaboration with Adidas for biodegradable sportswear. Why spider silk? Strength, 5x stronger than steel by weight. Flexibility, can stretch up to 40% without breaking. Biodegradable, reduces microfiber pollution from synthetic textiles. 4.1.2 Applications in fashion, medicine, and aerospace. Fashion, Stella McCartney debuted microbial silk dresses in 2023, showcasing cruelty-free luxury. Startups like Spiba, Japan, produce synthetic silk for outdoor gear. Medicine, silk scaffolds for tissue regeneration, for example, nerve repair. Antimicrobial sutures infused with engineered peptides. Aerospace, ultra-lightweight silk composites for satellite components. 4.2 Designer proteins and enzymes. Custom-engineered proteins and enzymes are revolutionizing industries by replacing toxic chemicals and enabling new manufacturing paradigms. 4.2.1 Industrial enzymes for sustainable manufacturing. Directed evolution. Companies like Novozymes and Codexis use CRISPR and AI to evolve enzymes for specific tasks. Example, Lacus enzymes break down lignin in plant biomass, enabling eco-friendly paper production. Key applications: biofuel production. Engineered cellulases convert agricultural waste into ethanol. Textile processing. Enzymes replace bleach in denim finishing, for example, evolved by nature's silk-based catalysts. Detergents. Proteases and lipases in Tide EcoBox clean stains at lower temperatures, reducing energy use. Environmental impact. Reduces reliance on petrochemicals and cuts co-emissions by up to 50% in manufacturing. Emerging innovations. Self-healing materials, microbes engineered to secrete proteins that repair cracks in concrete or coatings. Carbon capture enzymes. LanzaTech uses synthetic microbes to convert industrial emissions into ethylene. Challenges: cost. Scaling enzyme production remains expensive without breakthroughs in fermentation efficiency. Regulation. Novel biofabricated materials face strict safety reviews under EU's REACH and US EPA guidelines. Chapter 4 Summary. Synthetic biology transforms microbes into microscopic factories, producing everything from spider silk shirts to carbon-neutral jet fuel. Bold Thread's success exemplifies the fusion of biology and engineering, while designer enzymes redefine sustainable manufacturing. Yet, scaling these innovations requires tackling cost barriers and evolving regulations, a theme explored further in later chapters on biotech business models and global ethics. Chapter 5: Gene Editing and CRISPR Revolution. Gene editing, particularly through CRISPR-Cas9, has democratized the ability to rewrite life's code with unprecedented precision. This chapter explores CRISPR's transformative mechanisms, its agricultural and medical applications, and the ethical dilemmas it raises as humanity gains the power to redesign biology itself. 5.1 CRISPR-Cas9, Mechanisms and Applications. Mechanism: CRISPR-Cas9 is a bacterial immune system repurposed as a gene editing tool. Guide RNA: gRNA directs the Cas9 enzyme to a specific DNA sequence. Cas9 nuclease cuts the DNA, enabling deletions, insertions, or replacements. Repair: cellular machinery, NHEDG or HDR, fixes the break, allowing precise edits. Applications: basic research, rapidly creating gene knockouts in model organisms, for example, zebrafish, mice. Agriculture: engineering drought-tolerant crops, for example, Cibosu canola. Trademark symbol. Medicine: correcting genetic mutations in diseases like cystic fibrosis. Synthetic biology: rewriting microbial genomes for biofuel production. Case study: in 2023, the FDA approved Casgevy, Exocel, the first CRISPR-based therapy for sickle cell disease, developed by Vertex Pharmaceuticals and CRISPR Therapeutics. Next-gen tools: base editing, changes single DNA letters without cutting, for example, correcting APOE for Alzheimer's risk gene. Prime editing, search and replace edits with reduced off-target risks. 5.2 Agricultural innovations: disease-resistant crops. CRISPR is revolutionizing farming by addressing food security and climate challenges. Disease resistance: wheat, edited TAR-MLO genes confer resistance to powdery mildew, reducing fungicide use. Cassava: CRISPR-edited varieties resist brown streak virus, safeguarding a staple crop for 800 million people. Climate adaptation: rice, sub-1 gene edits enhance flood tolerance. Coffee: engineering caffeine reduction for easier decaffeination. Controversies: regulatory hurdles. The EU classifies CRISPR crops as GMOs, while the US and Japan exempt non-transgenic edits. Corporate control: critics warn of monopolies as firms like Corteva patent CRISPR-edited seeds. Case study: pairwise *S conscious greens. Trademark symbol: CRISPR-edited mustard greens with reduced bitterness, debuted in U.S. grocery stores in 2023, marketed as baby kale alternatives. 5.3 Therapeutic breakthroughs: sickle cell and beyond. CRISPR is rewriting the future of medicine, offering cures for previously untreatable genetic disorders. Sickle cell disease, SCD. Casgevy reactivates fetal hemoglobin by editing the BCL11 gene in hematopoietic stem cells, eliminating vaso-occlusive crises in 97% of trial patients. Cost: $2.2 million per treatment. Raises questions about equitable access. Beta thalassemia: CRISPR therapies boost hemoglobin production, reducing transfusion dependence. Beyond blood disorders: CAR T-cell therapy, editing immune cells to target cancers, for example, *CARVYKTI. Trademark symbol * for multiple myeloma. In vivo editing, Intellia Therapeutics NTLA 2001 reduces transthetin, TTR, levels in hereditary amyloidosis patients. Ethical Frontiers, Germline Editing, the 2018 Hygien QE scandal CRISPR-edited babies, sparked global calls for moratoriums. Equity, high costs and infrastructure gaps limit CRISPR therapies to wealthy nations. Chapter 5 Summary CRISPR has shifted biology from observation to engineering, offering solutions from famine-resistant crops to genetic cures. Yet, its power demands rigorous oversight. As agriculture and medicine embrace CRISPR, stakeholders must balance innovation with inclusivity, ensuring gene editing doesn't deepen global inequities. Later chapters delve into the business models and policies shaping this balance. Key takeaways: CRISPR's precision and affordability have democratized genetic engineering. Agricultural edits combat climate and disease but face regulatory fragmentation. Curative therapies like Casgevy mark a new medical era, yet accessibility remains a hurdle. Ethical frameworks must evolve alongside technical breakthroughs. Chapter 6: Biopharmaceuticals and Personalized Medicine. The convergence of biotechnology, genomics, and artificial intelligence is ushering in a new era of precision medicine, where therapies are tailored to individual patients' genetic profiles, lifestyles, and diseases. This chapter examines three pillars of this revolution: mRNA vaccines, CAR T-cell therapies, and RNA-driven drug discovery, innovations accelerated by the COVID-19 pandemic and poised to redefine healthcare. 6.1 mRNA vaccines, lessons from COVID-19. mRNA vaccines, once a fringe concept, became household names during the pandemic, demonstrating unprecedented speed and adaptability. Mechanism: mRNA vaccines deliver genetic instructions, via lipid nanoparticles, for cells to produce viral proteins, for example, SARS-CoV-2 spike protein, triggering immune responses without using live virus. Advantages: rapid design, weeks versus years for traditional vaccines, easy modification for variants. Case study: Pfizer /BioNTech and Moderna, speed to clinic. COVID-19 vaccines were developed in 1 year, leveraging decades of mRNA research. Scalability: Moderna's 2023 mRNA 1345 for RSV reduced infant hospitalizations by 83% in trials. Beyond pandemics, cancer: BioNTech's BNT111 targets melanoma via mRNA encoding tumor antigens. HIV: Moderna's mRNA 1644 induces broadly neutralizing antibodies in early trials. Challenges: cold chain logistics, mRNA's fragility requires ultra-cold storage, -20 degrees Celsius to -70 degrees Celsius. Public hesitancy, misinformation-fueled distrust, despite 95% efficacy rates. Legacy: the pandemic validated mRNA as a platform, with over 50 mRNA candidates now in trials for diseases from Zika to cystic fibrosis. 6.2 CAR T-cell therapy and cancer treatment. Chimeric antigen receptor T-cell, CAR T, therapy reprograms a patient's immune cells to hunt and destroy cancer. How it works: 1 T-cell extraction, blood is drawn, and T-cells are isolated. 2 Genetic engineering, a viral vector adds a CAR gene, enabling T-cells to target cancer-specific proteins, for example, CD19 in lymphomas. 3 Reinfusion, engineered cells multiply in the body, attacking tumors. Breakthroughs, approved therapies: Kymriah, Novartis' first CAR T approved, 2017, for pediatric acute lymphoblastic leukemia, ALL, achieving 83% remission. Yescarta, Gilead treats large B-cell lymphoma, with 40% long-term survival in refractory cases. Next-gen CAR-T, off-the-shelf CAR-T, Allogene Therapeutics Allo 501 uses donor cells, eliminating weeks-long customization. Solid tumors, trials target mesothelin, ovarian cancer, and HER2, breast cancer. Challenges, cytokine release syndrome, CRS potentially fatal immune overactivation, managed with tocilizumab. Cost, $375,000 per treatment limits access. 2023 milestone, CARVYKTI trademark symbol, Legend Biotech, doubled survival rates in multiple myeloma, earning FDA approval. 6.3 AI-driven drug discovery. Artificial intelligence is slashing drug development timelines from years to months, identifying targets and optimizing molecules with machine learning. How AI accelerates discovery, target identification. DeepMind's AlphaFold predicts 3D protein structures, 200M and cataloged, revealing new drug targets. Molecule design. In Silico Medicine's AI platform identified a fibrosis drug candidate in 18 months, versus 4-5 years traditionally. Recursion Pharmaceuticals screens millions of compounds virtually, prioritizing lab testing. Case studies. COVID-19. Benevolent AI identified baricitinib, an arthritis drug, as a COVID-19 treatment in days, later approved by the FDA. Rare diseases. Atomwise AI found a potential treatment for Ewing sarcoma in under 1 week. Challenges. Data quality. AI models require vast, clean datasets, often siloed in pharma archives. Validation. Many predicted drugs fail in clinical trials, for example, Oxycencr's DSP-1181 for OCD. Future trends. Generative AI, tools like ChatGPT for drug discovery draft molecular structures and trial protocols. Personalized drug cocktails, AI tailors combinations to individual tumor genomics. Chapter 6 Summary from NAS pandemic breakthrough to CAR-T's cancer conquests and AI's disruptive speed. Biopharmaceuticals are shifting medicine from one-size-fits-all to bespoke solutions. Yet, challenges, cost, access, and validation, loom large. As personalized therapies become mainstream, stakeholders must ensure these innovations don't deepen healthcare inequities but instead fulfill their promise: the right treatment, for the right patient, at the right time. Key takeaways: mRNA, a versatile platform for rapid response vaccines and personalized cancer therapies. CAR-T, a paradigm shift in oncology, though toxicity and cost hinder scalability. AI, redefining drug discovery but requiring rigorous real-world validation. Ethics, balancing profit motives with equitable access remains critical. Next frontiers, combining mRNA and CAR-T for in vivo cell engineering. AI-designed gene therapies for ultra-rare diseases. Global partnerships to democratize access, for example, whose mRNA tech transfer hubs. Chapter 7, Environmental Biotechnology. Environmental biotechnology harnesses biological processes to address pressing ecological challenges, offering innovative solutions to pollution, climate change, and waste. This chapter explores 3 transformative approaches: microbial remediation, algae-based systems, and sustainable packaging, each leveraging nature's ingenuity to foster a greener future. 7.1 Microbial remediation of pollution. Microorganisms are deployed as environmental custodians to detoxify polluted ecosystems. Oil spills. Alcanivirax borcumensis bacteria metabolise hydrocarbons, notably used in the Gulf of Mexico post-Deepwater Horizon. Heavy metals. Genetically modified Deinococcus radiojurans absorbs radioactive waste, while Geobacter species reduce uranium contamination in groundwater. Plastic degradation. CRISPR-engineered Ideonella saccaiensis breaks down PET plastics, achieving 90% depolymerization in lab settings. Challenges: scalability, public skepticism toward GMOs, and regulatory hurdles for field applications. 7.2 Algae-based biofuels and carbon capture. Algae's rapid growth and CO2 absorption make it a dual-purpose tool for energy and climate mitigation. Biofuel production. Companies like Algenol optimize algae strains for high lipid yields, converting CO2 into ethanol. ExxonMobil's research aims for commercial viability by 2030. Carbon sequestration. Brilliant Planet uses algal blooms to capture 30 tons of CO2 per hectare annually, storing carbon in biomass for agricultural use. Wastewater integration. Startups like Algae Systems treat sewage while producing biofuel feedstock, closing resource loops. Challenges: energy-intensive harvesting and cost competition with fossil fuels, $7.9 per gallon versus $3 for diesel. 7.3 Sustainable packaging solutions. Biotech pioneers are replacing plastics with compostable, bio-based materials. Mycelium packaging. Ecovative Design grows mushroom-based materials for IKEA and Dell, offering fire-resistant, biodegradable alternatives to polystyrene. Seaweed innovations. Not Pla's edible seaweed pods, used in leukosid sport, decompose in 4-6 weeks, avoiding microplastic pollution. Plant-based polymers. Mango Materials produces PHA from methane waste, used in rigid packaging and textiles. Challenges: cost. Bioplastics are 2-3x pricier than conventional plastics, and durability under varied conditions. Chapter summary. Environmental biotechnology merges microbiology, synthetic biology, and materials science to tackle pollution, energy, and waste. From oil-eating bacteria to algae carbon farms and mushroom packaging, these innovations exemplify nature-inspired solutions. Yet, scalability, cost, and public acceptance remain barriers. With global bioplastics markets projected to reach $34 billion by 2030 and algae biofuels poised to offset 12% of aviation emissions, the sector's potential is vast, if supported by policy, investment, and interdisciplinary collaboration. As climate urgency grows, these biotech tools underscore a critical truth: the solutions to Earth's greatest crises may lie in its smallest organisms. Key takeaways: microbial remediation offers targeted cleanup but faces GMO stigma. Algae systems dualize energy production and carbon capture, needing cost breakthroughs. Sustainable packaging reduces plastic waste but competes with entrenched industries. Future success hinges on scaling technologies, circular economy integration, and public engagement. Next frontiers: CRISPR-enhanced microbes for forever chemical degradation. Algae-integrated urban infrastructure, for example, biofacades for buildings. Policy incentives like carbon pricing to accelerate adoption. Chapter 8: Regulatory and ethical challenges. Biotechnology's rapid advancement often outstrips the frameworks designed to govern it, creating tensions between innovation, safety, and equity. This chapter examines the regulatory labyrinths, biosecurity risks, and access disparities shaping the ethical landscape of biotech. 8.1 FDA, USDA, and global oversight frameworks. Regulatory bodies struggle to balance safety with innovation in fast-moving fields like gene editing and cellular agriculture. U.S. agencies: FDA, oversees biopharmaceuticals, gene therapies, and lab-grown meat. For example, 2023 approval of Upside Foods cultivated chicken. USDA regulates genetically engineered crops, for example, CRISPR-edited non-browning mushrooms exempt from GMO labeling under 2020 secure rule. EPA monitors bioengineered microbes released into the environment, for example, Lanzatecas CO2 converting bacteria. Global frameworks: EU, strict GMO laws require pre-market approval for CRISPR crops, stifling startups. The novel food regulation delayed Eat Just's cultivated meat entry until 2025. China fast-tracks gene-edited crops to ensure food security, for example, high-yield CRISPR rice approved in 2022. Africa: the African Union's model law on biosafety promotes cautious adoption, with Kenya approving Bt cotton in 2023. Challenges: fragmented regulations create market barriers, for example, Calixt's gene-edited soy blocked in the EU, and slow crisis responses, for example, delays in approving mRNA vaccines during COVID-19. 8.2 Biosecurity and dual-use research concerns biotechnology's dual-use potential, for both curing diseases and engineering pathogens, demands urgent safeguards. Key risks: synthetic pathogens. In 2017, researchers controversially recreated the extinct horsepox virus, raising fears of smallpox synthesis. Gene drives, engineered to eradicate malaria mosquitoes, but potential ecological disruption led to a 2023 UN moratorium on field trials. RNA-driven bioengineering. Tools like Meta's ESM-Fold could be misused to design toxic proteins. Mitigation strategies: screening DNA orders. The International Gene Synthesis Consortium * screens orders for harmful sequences. Biosecurity certifications. NIH now requires grants involving enhanced pathogens to pass NSAB review. Whistleblower policies. Boston University's 2022 gain-of-function flu study sparked calls for transparent risk-benefit assessments. Case study: the World Health Organization's 2023 guidance on AI for pandemic prediction * emphasizes ethical use to prevent weaponization. 8.3 Equity in access to biotech innovations. Biotech breakthroughs often bypass marginalized populations, exacerbating global inequities. Access barriers. Cost. Casgevy, the $2.2 million CRISPR therapy for sickle cell disease, remains inaccessible in sub-Saharan Africa, where 75% of cases occur. Patents. Modernizing medicine * S patent on RNA-driven drug discovery algorithms limits global South access. Infrastructure gaps. mRNA vaccines require cold chains. Only 35% of African health facilities have reliable refrigeration. Equity initiatives. Tech transfer hubs, whose mRNA hub in South Africa trains scientists from 15 nations to produce local vaccines. Open source platforms. BioBricks Foundation shares genetic tools royalty-free for non-commercial use. TID pricing. Novartis offers CAR-T therapy Kymriah at reduced costs in India and Brazil. Case study. Cuba's Center for Genetic Engineering and Biotechnology produces affordable cancer vaccines, for example, Symavac CGF, despite U.S. sanctions. Chapter summary. Biotechnology's promise is tempered by regulatory fragmentation, biosecurity risks, and systemic inequities. While the FDA and USDA navigate CRISPR crops and lab-grown meat, dual-use research and AI demand global governance. Meanwhile, initiatives like WHO's mRNA hubs and open-source biology strive to democratize access. The path forward requires harmonizing innovation with inclusivity, ensuring biotech serves humanity, not just the privileged few. Key takeaways: regulatory divergence, US agility versus EU caution creates market imbalances. Dual-use dilemmas, balancing scientific freedom with preemptive safeguards. Equity gaps, high costs and patents hinder Global South access to life-saving tech. Solutions, tech transfers, TID pricing, and ethical AI frameworks. Future directions, global treaties to standardize biotech oversight, for example, biological CRISPR protocol *. Ethical IP models requiring patent holders to subsidize low-income access. Public-private partnerships for decentralized biomanufacturing, for example, modular mRNA labs. Chapter 9. The Business of Biotechnology 9.1 Startups versus Giants. Key players in the bioeconomy. The biotech sector thrives on a dynamic interplay between agile startups and established giants, each driving innovation in distinct ways. 9.1.1 Bold Threads, Impossible Foods, and Moderna. Bold Threads, a startup pioneering biofabrics, such as Microsilk trademark symbol, engineered spider silk, and MLO trademark symbol, mycelium leather. Partnered with brands like Stella McCartney and Adidas to commercialize sustainable materials. Impossible Foods, known for plant-based meat alternatives, it leverages heme, soy hemoglobin, to mimic meat's taste and texture. Positioned as a disruptor in the $1.40 global meat market. Moderna transitioned from a startup to a biotech giant via its mRNA platform. Post-COVID-19, it expanded into oncology, for example, mRNA 4157/V940 cancer vaccine, and rare diseases, showcasing scalability of mRNA tech. Contrast, startups like Bold Threads focus on niche, sustainable materials, while giants like Moderna dominate therapeutic markets with deep R&D pockets. 9.1.2 Venture capital trends in biotech. 2023 investment surge. Biotech VC funding reached $34.6 billion, with synthetic biology startups raising $5.3 billion, CB Insights. Hot sectors, alternative proteins. Companies like Upside Foods, $400 million Series C, and Perfect Day, animal-free dairy, attract climate-focused investors. Biofabrication, Ecovative Design secured $60 million for mycelium materials. Impact investing, ESG-driven funds prioritize startups with circular economy models, such as MycoWorks, Mushroom Leather, and LanzaTech, carbon recycling. 9.2 Market potential of lab-grown meat and biofabrics. Lab-grown meat, valuation, projected to hit $25 billion by 2030, McKinsey, driven by environmental concerns. Lab meat reduces GHG emissions by 92% versus beef. Key players, Upside Foods, USDA-approved chicken, and Masa meat, €85 million raised in 2023, lead commercialization. Challenges, scaling bioreactors and slashing costs, currently $50 per cultured burger versus $5 for conventional. Biofabrics, market growth, expected to reach $3.4 billion by 2030, fueled by fashion sustainability demands. Innovations, Bold Threads, collaborates with luxury brands to mainstream low trademark symbol. SPBA, Japanese firm producing synthetic silk for North Face jackets. Hurdles: achieving cost parity with polyester, $1.50/kilogram versus $5/kilogram for spider silk. 9.3 Public perception and marketing strategies. Public perception: lab-grown meat, 35% of U.S. consumers are willing to try it, citing environmental benefits, Pew Research. However, 48% express safety concerns. Biofabrics: 60% of millennials prefer sustainable materials, but lab-grown labels can deter traditionalists. Marketing strategies: transparency, Upside Foods offers facility tours to demystify production. Sensory appeal: Impossible Foods emphasizes taste via campaigns like the meat of plants. Celebrity partnerships: Bold Threads collaborates with Stella McCartney to position biofabrics as luxury. Education: Good Meat uses documentaries to highlight ethical and climate benefits. Challenges: greenwashing risks: companies face backlash if sustainability claims lack third-party verification, for example, B Corp certification. Regulatory hurdles: the EU's novel food label for cultured meat may slow adoption versus the U.S. cell-cultivated branding. Chapter summary: the biotech business landscape is a battleground of innovation, where startups like Bold Threads disrupt industries with sustainable alternatives, while giants like Moderna scale cutting-edge therapies. Venture capital fuels sectors from lab-grown meat to biofabrics, yet challenges in cost, scalability, and public trust persist. Success hinges on strategic marketing, balancing transparency, sensory appeal, and ethical storytelling, to convert skepticism into adoption. As the bioeconomy expands, collaboration between startups, investors, and regulators will determine whether biotech delivers on its promise of a sustainable and equitable future. Key takeaways: startups drive niche innovations, giants scale solutions with global impact. Lab-grown meat and biofabrics face cost and perception hurdles despite immense market potential. Effective marketing blends education, transparency, and partnerships to overcome consumer skepticism. The future bioeconomy will hinge on sustainable practices, ethical branding, and regulatory agility. Chapter 10: Future Horizons, 2030 and beyond. By 2030, biotechnology will transcend current limitations, merging with AI, nanotechnology, and climate science to tackle humanity's grand challenges. This chapter envisions a future where bioprinted organs save lives, programmable microbes reverse ecological damage, and AI-driven bio-nanosystems redefine what's possible. 10.1 Bioprinting organs and tissues. Bioprinting promises to end organ transplant shortages by fabricating living tissues layer by layer. Advances: vascularization breakthroughs, volumetric bioprinting, 2025 3D printed liver tissues with integrated blood vessels, developed by Prelis Biologics, achieve functional perfusion in animal trials. Organovos kidney patches, 2027 printed renal tissues reduce dialysis dependency in chronic kidney disease patients. Materials innovation, cell-laden bioinks, hybrid hydrogels with graphene enhance electrical conductivity for heart tissue printing. Decellularized scaffolds, repurposed plant cellulose, for example, spinach leaves, structure human capillaries. 2030 milestones: first bioprinted human heart transplant, pending FDA approval. Bioprint on demand * clinics for cartilage and skin grafts. Challenges: immune rejection of synthetic tissues. Scalability for complex organs, for example, lungs. 10.2 Programmable microbes for climate solutions. Engineered microbes will become frontline tools against climate change, converting waste into resources and capturing carbon. Key innovations: CO2 to fuel microbes, LanzaTech's air carbon. 2025 Bacteria convert industrial emissions into ethylene at commercial scale, displacing fossil-derived plastics. Synthetic genomics algae strains, 2026 absorb CO2 20x faster than natural varieties, producing biocement for construction. Methane mitigation, Archon Biotechnologies, 2027 Archaea microbes convert CO2 into protein-rich food, reducing livestock methane demand. 2030 Vision, Global Carbon Recycling Network, microbe-powered plants capture 1 gigaton of CO2 annually, integrated with carbon credit markets. Challenges: ecological risks of engineered microbes in open environments. Public resistance to GM ecosystems. 10.3 Convergence of AI, nanotech, and biotechnology. The fusion of AI, nanotech, and biotech will create programmable biology, a new frontier of precision and control. Synergistic applications: AI-designed nanobos, DNA origami nanorobots. 2028 AI-optimized structures deliver CRISPR payloads to cancer cells, leaving healthy tissue untouched. Sensoria Therapeutics Neural Dust. 2029 Nanoparticles monitor neurotransmitters in real time, treating epilepsy via closed-loop feedback. Synthetic biology platforms, Ginkgo Bioworks cell development kits. 2030 AI-guided biofoundries print custom microbes for pharma, agriculture, and materials. Ethical frontiers, privacy risks. Nanobos collecting biometric data could enable surveillance. Autonomous systems, AI-controlled bio-nanofactories raise dual-use concerns, for example, bioweapon synthesis. Chapter 10 Summary: The 2030s will blur the lines between biology and technology. Bioprinted organs could render donor wait lists obsolete, engineered microbes might reverse centuries of emissions, and our nanotech hybrids could cure diseases at the molecular level. Yet, these leaps demand rigorous safeguards, ethical frameworks, equitable access, and global collaboration, to ensure biotechnology elevates humanity without compromising our values. As these tools converge, they challenge us to redefine life itself. Key takeaways: bioprinting, focus on vascularization and immune compatibility to achieve transplant-ready organs. Programmable microbes, balance climate impact with ecological caution. Convergence, AI and nanotech amplify biotech's potential but require unprecedented governance. Ethics, global treaties must address autonomy, privacy, and equity in emerging bio-nanosystems. Beyond 2030, living buildings, bioengineered coral reefs and algae phakids regulate urban temperatures. Human-machine symbiosis, neural nanobos enhance cognition and treat neurodegeneration. Interplanetary biotech, microbial terraforming and printed habitats for Mars colonization. Chapter 11, Conclusion. Balancing innovation and responsibility. Biotechnology stands at a crossroads. Its power to heal, nourish, and sustain is matched only by its potential to deepen inequities, destabilize ecosystems, and challenge ethical norms. As this book has charted, from CRISPR's precision to bioprinted organs and AI-driven drug discovery, the field's trajectory hinges on harmonizing audacious innovation with unwavering responsibility. This final chapter distills lessons from preceding sections into a roadmap for a future where biotechnology uplifts all life on Earth. 11.1 Ethical Frameworks for Emerging Technologies. The speed of biotech innovation demands ethics that evolve as dynamically as the science itself. Adaptive governance, CRISPR and germline editing *. Building on Chapter 5's lessons, global consensus is needed to ban human germline edits until safety, equity, and oversight are assured. The WHO's Human Genome Editing Registry, 2023, offers a model for transparency. Dual-use risks, as synthetic biology converges with AI, Chapter 10, treaties akin to the Biological Weapons Convention must address are designed pathogens. Inclusive deliberation, citizen assemblies, like France's Convention Citoyen pour le Climat, could guide policies on gene drives and lab-grown meat, Chapters 3 and 7. Case study: the Global Biodata Alliance's 2025 framework balances open science with biosecurity, requiring data sharing agreements for sensitive genomic research. 11.2 Educating the next generation of biotechnologists. Future innovators must wield technical mastery alongside ethical literacy. Interdisciplinary curricula, programs blending biology, AI, and ethics, for example, MIT's Biological Engineering and Society degree, prepare students to navigate dilemmas like CRISPR equity, Chapter 5, and bioprinting access, Chapter 10. USCO's Bioethics Education Initiative * trains Global South scientists in responsible innovation. Mentorship and representation, initiatives like Black in Biotech and 500 Women Scientists diversify leadership, ensuring solutions reflect global needs, for example, sickle cell therapies in Chapter 6. Industry-academia partnerships, for example, Ginkgo Bioworks internships, bridge theory and real-world impact. Case study: Rwanda's African Institute for Mathematical Sciences integrates biotech and AI, empowering graduates to tackle local challenges like malaria-resistant mosquitoes. 11.3 Global Collaboration for Equitable Progress. Biotechnology's benefits must transcend borders, avoiding the pitfalls of past technological divides. Equitable partnerships, tech transfer hubs, expanding the WHO's mRNA vaccine model, Chapter 6, to CRISPR therapies and biofabrics, Chapter 4. Patent waivers, following COVID-19 precedents, critical tech, for example, drought-resistant CRISPR crops in Chapter 5, could be licensed royalty-free for low-income nations. Climate Solidarity, Global Bioeconomy Alliance, a proposed coalition to scale algae carbon capture, Chapter 7, and microbial remediation in the Global South. Loss and Damage Fund, directing a portion of biotech profits, for example, synthetic spider silk in Chapter 4, to nations disproportionately impacted by climate change. Case study: Cuba's Finlay Institute, despite embargoes, partners with Iran and Vietnam to produce affordable meningitis vaccines, exemplifying South-South collaboration. Final synthesis: Frontiers of Space Technology, Innovations and Beyond began by framing biotechnology as a tool to redefine life itself. It concludes with a call to redefine progress, one measured not by patents or profits, but by justice, sustainability, and shared dignity. The CRISPR cures, lab-grown meat, and carbon-capturing microbes detailed in these pages are not inevitabilities, they are choices. Choices to prioritize inclusive innovation over exclusionary markets, precautionary ethics over reckless speed, and planetary stewardship over short-term gain. As we engineer the future, let us encode it with the wisdom of Asilomar, Chapter 2, the urgency of pandemic responses, Chapter 6, and the creativity of biofabrics, Chapter 4. Let us build a world where biotechnology's boldest applications serve not the few, but the many, and where the next generation inherits not just advanced tools, but a moral compass to wield them wisely. In the words of Dr. Jane Goodall, what you do makes a difference, and you have to decide what kind of difference you want to make. Biotechnology's difference begins now. End of book. Appendices Appendix O, Timeline of Key Biotech Milestones, 2000-2023, 2000, Completion of the Human Genome Project Draft, Revolutionizing Genetics Research. 2003, First synthetic virus, polio, created by Eckhard Wimmer sparking debates on synthetic biology ethics. 2012, CRISPR-Cas9 gene editing system discovered by Jennifer Doudna and Emmanuel Charpentier. 2015, FDA approves Amgen's CRISPR-edited CAR T-cell therapy for leukemia. 2017, Perfect Day debuts animal-free dairy proteins using microbial fermentation. 2020, mRNA vaccines, Pfizer-BioNTech and Moderna, authorized for COVID-19, marking a pandemic breakthrough. 2021, CRISPR Therapeutics initiates first-in-human trials for exocell, sickle cell therapy. 2022, Bold Threads scales production of microbial spider silk for commercial fashion lines. 2023, USDA grants approval to Upside Foods and Good Meat for cultivated chicken sales in the U.S. Appendix B, Glossary of Technical Terms, CRISPR-Cas9, a gene editing tool derived from bacterial immune systems, enabling precise DNA modifications. mRNA vaccine, a vaccine using messenger RNA to instruct cells to produce antigens, triggering immune responses. Bioreactor, a controlled environment for growing cells or tissues, for example, for lab-grown meat or microbial silk. Synthetic biology, designing and constructing biological systems for new functions, for example, engineered microbes. Phage therapy, using bacteriophages, viruses, to target antibiotic-resistant bacterial infections. Biofabrication, producing materials, for example, leather, textiles, using living organisms like fungi or bacteria. CAR T therapy, genetically modifying a patient's T cells to attack cancer cells. Appendix C, Profiles of leading biotech firms and innovators. Companies. Moderna, pioneer of mRNA technology, expanded from vaccines to cancer and rare disease therapies post-COVID. CRISPR Therapeutics, co-founded by Emmanuel Charpentier, developed Casgevy, the first FDA-approved CRISPR therapy. Bold Threads, innovator in biofabrics, known for Microsilk trademark symbol, spider silk, and MLO trademark symbol, mycelium leather. Upside Foods, leader in cultivated meat, achieving USDA approval for lab-grown chicken in 2023. Ginkgo Bioworks, synthetic biology powerhouse offering RNA-driven organism design for industries from pharma to agriculture. Innovators, Jennifer Doudna, co-inventor of CRISPR-Cas9, Nobel laureate and advocate for ethical gene editing policies. Emmanuel Charpentier, CRISPR pioneer, co-founder of CRISPR Therapeutics and *ERS Genomics. Patrick O'Brown, founder of Impossible Foods, revolutionizing plant-based meat with hemp protein. Derek Rossi, co-founder of Moderna, catalyzed the mRNA vaccine revolution. Lei Xu, founder of Bota Biosciences, engineering microbes for sustainable chemical production. Purpose of appendices. These sections provide quick reference tools to contextualize the book's themes. The timeline anchors breakthroughs in historical progress. The glossary demystifies jargon for readers across disciplines. Profiles spotlight key players driving biotechnology's evolution, from lab benches to global markets. End of appendices.